Drosophila has been incredibly important as a model organism because it's been obviously very central to the development of modern genetics, and that's where it came from. But it then also became a very familiar organism because the guys who developed Drosophila genetics in Thomas Morgan's lab, they studied the life cycle of this fly from the beginning to the end. They looked carefully for all sorts of mutants in all different types, behavioral mutants, physiological mutants, anatomical mutants. So they came to know this fly incredibly well.
Yeah.
So this may explain why it made it into all sorts of fields of biology which aren't necessarily that closely related to genetics. They may well be closely related to genetics, but they're not necessarily closely related to genetics. The main reason is that these people came to know this fly so well. So there's no way of telling whether it's actually a better model than any other organism. It's just the one organism that people got to know so well. I think ethics has become much more important in recent years than it was at the beginning. Obviously you had concerns about the use of laboratory animals already in the 19th century. And the British were particularly concerned about the use of animals. There was a strong anti-vivisectionist movement in Britain, and a lot of the experiments that were possible and common in France and in Germany weren't accepted, or were at least very controversial in this country. But this, I think, was not important for Morgan's choice of Drosophila as a research organism. Now, since the Second World War, this has become far more regulated, and it's much easier to organize research with organisms that are not dogs or cats, but little flies or bacteria where nobody attaches much emotional value to the animal. Well, it's hard to tell, but I would predict that it becomes that it stays a very important research organism, simply because there's a certain path dependency in this. You have a lot of people working with Drosophila already. You have a lot of knowledge about the Drosophila genome and the phenome, if you want. As there are new problems that people deal with, there will always be somebody who says, why don't we do this in Drosophila? We know how to deal with Drosophila, why don't we look at this in Drosophila? Well, it depends on what you were doing, but already one important field of research in the 19th century was nerve function. A lot of people were interested in nerve function. I think we didn't talk about it because this lecture was sacrificed to the reading week. Had we talked about this, we would have seen that at the beginning the French were doing a lot of that research on dogs and cats, and the problem there was that these animals were so obviously suffering extremely badly that the results of the research were open to all sorts of challenges. You know, if you do something on nerves and sensibilities, if you have an animal that suffers incredibly badly, this may not be adequate research results. And that's when people like Johannes Müller, about whom we've talked a lot, moved on to the frog. And you then find that a lot of the research on nerves and on muscle-nerve interactions, on the whole electricity of the body, is done in frogs. So frogs, I think, you could view frogs as one model animal of the 19th century, but dogs and cats and rabbits remained quite central. It always depends on what you're doing. There was a tool for every job, and some animals sort of became the standard animals in some fields and others in other fields. Guinea pigs and rabbits were quite central for bacteriological research. Mice became extremely central for cancer research. So you have different animals in different fields.
Yeah.
I mean, a frog obviously wouldn't be a suitable model organism to study drug efficacy on humans. It wouldn't be right. So you simply couldn't use it. You wouldn't use a frog. You would use, and again, this is these are difficult questions. You have to use an animal that's sufficiently similar to humans, but that is still within what's ethically possible, and that's the kind of difficulties you're facing. It's difficult. It's difficult. I'm thinking about sort of important episodes in the history. I mean, you're talking about human drug development for human use. An important episode and a sort of tragedy of post-war drug development, pharmaceutical research, was the thalidomide scandal. I don't know if that's thalidomide, this drug which caused nerve problems in adults, but the main big scandal involved birth defects in newborns, and which were, I mean, pre-birth in the early early embryonic development. The limb development didn't happen properly. Now, this was not properly tested for anywhere before thalidomide. Before the thalidomide scandal blew up. So it's not that there isn't a good animal model, but if you don't know what you're looking for, you may not have it in your repertoire.
Yeah.
And after thalidomide, people looked actively for animal models that may represent these, that may pick up these pre-birth these birth defects and these these disturbances of embryo development. But it's not due to. I mean, then there's an active search for a good animal model, and you may find one, or you may not find one, but you know exactly what you're looking for. Cheap, reproduces quickly, easy to keep in the lab. Lots is known about, lots of knowledge about it.
Yeah.
I think.
And been central to biology ever since. Well, I think that's great. Thank you.
Thank you.
A lot.